This presentation focuses on the role of vaccines in pandemic response. We will talk through four main themes. Firstly, what can vaccines do to reduce harms in a pandemic? What evidence will we have about pandemic vaccines to know that they're safe and effective? And when will we know this? We'll talk about two types of vaccine protection, direct and indirect. And we'll talk about the trade-offs between what I'm calling partial and complete vaccination. If 2 doses of vaccine are needed to fully protect against infection, is it better to give more people a single dose with partial protection or fully immunise a smaller group of people? But first, let's begin by reviewing how flu vaccines work. The vaccines that are available to us in Australia are what we call killed flu vaccines. They're made from influenza viruses that are grown and then purified so that we can take just the antigens or the outer components of the virus that generate an immune response and put these together in a vaccine formulation. Typical seasonal influenza vaccines need to be able to cover the viruses that circulate every year in the human population. 2 main groups of flu viruses circulate in humans, influenza A, and of these there are 2 subtypes, H3 and H1 viruses, so we need a representative of each of those in the vaccine, and also influenza B. Influenza vaccines have typically contained 1 B virus strain, but some more recent vaccines contain 2 B virus strains, so they have either 3 or 4 antigens each year. Influenza vaccines evolve year on year, and so the vaccines that are provided need to be updated and only protect for 1 year. We're representing the process by which those viruses evolve through the cartoon on the right. The first virus there has antigens on the surface represented as yellow triangles. Through a process of gradual evolution and antigenic drift, each year's viruses are slightly different, and so we see the virus on the top that has yellow triangles of a different shape from the preceding year. Individuals are unlikely to be protected against that virus by last year's vaccine. Influenza vaccines produce antibodies that come into contact with the virus on initial exposure and neutralise it on entry, preventing the virus from growing within an individual. These vaccines are available globally and updated twice each year, once for the Northern Hemisphere season and once for the Southern Hemisphere influenza season. By definition, a pandemic virus is more different from previously circulating viruses than the one we saw last year, and that's why more people are vulnerable or susceptible to it. And this degree of difference is reflected in the cartoon again, looking at the virus on the lower right, which is a pandemic virus that's arisen by a process of antigenic shift. So a new group of antigens has come into the population, usually by transfer from birds. And the difference is they're represented in the cartoon by the red squares. So we said that flu vaccines neutralise the virus on entry, and this means that they protect against the whole course of infection. When we think about influenza infections in the population, the ones that we see and the ones that occur, we talk about a pyramid of severity. We know from studies that maybe 1/3 of people who have evidence of growing influenza virus will have no symptoms at all. Others may feel unwell at home or even go to work, but we can only record and identify through the public health system those people who actually seek medical care by attending a GP, being admitted to hospital, requiring intensive care admission, or even dying. So we assume that vaccination can prevent this entire pyramid, but the only portion of it that we can actually identify and measure is the proportion of people who seek clinical care. We talk about vaccines as being a certain percent protective or effective against disease. And I just want to clarify exactly what we mean. This schematic represents the the idea of the proportion protected. It's not based on any real numbers that are relevant to flu, it's purely here to make a point. On the left-hand side of the figure, we represent 12 individuals who have not received any vaccine and are not protected against disease. On the right-hand side, we have a group of people who've received a vaccine that is 50% protective. This means that each individual on exposure to flu has a 50% reduction in the risk of going on to develop an infection. So, for example, if people on the left were all exposed to influenza, we would anticipate that 4 of them would go on to develop infection. If a vaccine is 50% protective, then the risk of that happening is reduced by half each time, and only 2 people would develop the infection after exposure. And it's this kind of assessment that allows us to estimate the percent protection or the percent efficacy of a vaccine. So what's the evidence that we have that flu vaccines protect? Influenza vaccines have been available for many decades and trialled for many decades in different ways. There are 2 types of studies that provide clear evidence that flu vaccines reduce the risk of infection after exposure. The first series of studies are human challenge studies, and the most famous were undertaken at the Common Cold Unit in Salisbury in the United Kingdom. This centre recruited willing volunteers to come and spend their holidays staying in little huts out in the countryside. Individuals would have their antibody levels to influenza measured, some would be vaccinated, others would not, and they would then be exposed to influenza or, in related studies, other cold and respiratory viruses. This rather glamorous couple are staying in their unit and being visited by one of the workers from the facility who is cunningly protected from being infected by them or bringing an unwanted infection to them. And these studies gave good evidence that antibodies related to vaccine could protect. Randomised controlled trials are a larger scale type of study, and these randomly allocate people to either receive a vaccine or not. And in this case, they usually require thousands or tens of thousands of people to be involved in the studies. Individuals having been assigned to groups are followed over time. Disease cases are identified and counted in both groups, and the rates of disease in each of the vaccinated and unvaccinated groups are compared to calculate protective efficacy. So these studies are highly effective. They give us a strong evidence basis that vaccines work, but they are time-consuming and costly, and there is certainly no time to undertake this level of rigorous scrutiny of a vaccine in a health emergency, because by the time you have conducted your study, the whole thing will be over. So we need to be able to predict whether or not a new vaccine is likely to protect. Those human challenge studies that we talked about in Salisbury were really useful because they directly related the antibodies in individuals to their risk of contracting the flu after exposure. That means that we've been able to define vaccine-associated antibody levels that substantially reduce the risk of disease. So we're able to just measure antibody responses and use these as a proxy or a correlate of protection. Studies like this can be conducted quite rapidly because we don't have to hang around waiting for people to get infected. And they need to be conducted in both adults and children because we predict, based on studies, that antibody responses will be different by age, based on people's track record of having been exposed to flu viruses over the course of their lifetime. These studies are also pivotal to identify whether one dose of vaccine will be sufficient to generate antibody levels high enough to protect, or whether 2 doses will be needed. We're able to go through this process every year for seasonal flu vaccines. And the process has been streamlined from a regulatory perspective because the basic vaccines used remain the same, and all that's updated each year is the flu virus antigens. Manufacturers are able to check antibody responses in small groups of individuals, and each year we need to monitor vaccine safety in the population to be sure that even this minor change does not change the vaccine safety profile. Licensing pandemic vaccines builds on this year-on-year seasonal experience. Again, the same vaccine backbone may be used to generate a pandemic vaccine. There are other types of vaccines that knowingly stimulate higher immune responses that have also been used in clinical trials and are pre-approved in many countries. Pandemic vaccines are a bit simpler to make in that only one virus antigen needs to be incorporated rather than the usual 3 or 4. And rapidly conducted clinical trials will be undertaken in a pandemic to assess antibody responses and get some initial idea of vaccine safety before the vaccines are given to the healthy human population. It's important to note that these trials will involve maybe a few hundred people, so they will be able to detect common side effects like a bit of redness or soreness at the injection site or sometimes fevers, but sometimes vaccines can cause rare but important side effects, maybe in 1 in 1,000 or 1 in 10,000 people, that won't be able to be detected by these early trials. So it's important that we keep monitoring what's happening with safety once a vaccine has been given to large numbers of people. So how do we know if the vaccine's working? So here we need to conduct what we call observational studies of following what happens when the vaccine is used in the real world. Some people will become vaccinated, others will not, either because they choose not to receive the vaccine or because they're not eligible for it. We will have surveillance systems in place to see who gets disease and can use those to compare vaccinated and unvaccinated people. And there are a number of study designs that can be done to gather this information, looking at institutional outbreaks, comparing people identified as cases with others who haven't had the disease, or by following large groups of people whose vaccine status is known and waiting for cases to be identified. By comparing the rates or odds of disease in both groups, we can calculate the protective effectiveness of the vaccine, which is the measure of how well the vaccine protects in a real-world setting. And by comparing the overall reduction in disease from what might be expected in settings where vaccine is not used, we get a measure of the overall impact of the vaccine in reducing the burden of disease. We talked about the importance of monitoring vaccine safety, and in a country like Australia, we have multiple vaccine safety systems in place. The government has a passive adverse events reporting system for all drugs, including vaccines. Drug companies are also mandated to gather information on adverse side effects of vaccines, and we have clinic networks around the country that are focused on vaccine safety, which record adverse events and regularly discuss these to identify any unexpected vaccine signals. There have also been some game changers in vaccine safety monitoring in recent years, including an app called SmartVax, which has been implemented in a number of GP practices around the country to monitor influenza vaccine safety. Using electronic health records, individuals who've received the vaccine can be identified. A day or two after vaccination, they're sent an SMS asking whether they would like to be part of the study and also identify whether or not they have had any side effects related to the vaccine. If they report that they have, these will be followed up in more detail. But in this way, and using an electronic system outside our usual record systems, we've been able to rapidly gather information on tens of thousands of people within a matter of weeks of flu vaccine introduction each year. When we use vaccines in the population, there are different strategies that we can use to try to reduce disease. And there are two main types of protection that we're going to discuss here. Those are direct and indirect protection. Vaccines are different from other medications. If you receive a blood pressure medication, it will lower your blood pressure, but it won't have any effect on anybody else. If you receive a vaccine, it can prevent you from getting an infection, and as a consequence, you are then unable to infect other people, and that's an indirect benefit to the people around you. The schematic in the figure represents this phenomenon in 3 levels, depending on the degree of vaccination in the population. In the top schematic, all of the people in blue are not immunised but still healthy, but they could contract an infection. As red infected individuals move into the population, they find lots of people around them susceptible to infection, and the disease can spread rapidly through the population. In the middle cartoon, some people have been immunised, but only a small proportion. Those people are directly protected from infection themselves, but because there are still a lot of vulnerable or susceptible people around in blue, they are still able to become infected, and the disease will spread widely. If, however, we can immunise a very large proportion of the population, and here in the bottom figure we see a lot of yellow people on the left, then they effectively act as a roadblock to the spread of disease, as most of the people that infected individuals come into contact with can't be infected. In this situation, the disease does not take off and spread in the population, and this level of immunisation is something that we strive for in some immunisation programs to produce what's called herd immunity as a population level benefit of this indirect protective effect. Who needs to be directly protected by vaccines? So each year, the Australian government releases an influenza statement, and the government decides to pay for influenza vaccines for groups of people who are at risk of more severe disease than others. In 2019, seasonal influenza vaccines are being provided freely through the National Immunisation Program for all Aboriginal and Torres Strait Islander people aged more than 6 months, for pregnant women, for all people aged over 65 years, and for people over 6 months with conditions which increase flu complications like heart or lung disease. However, we need to be mindful that in a pandemic, different groups of people might be observed to be at higher risk of severe disease than in a usual influenza season. And so studies will be put in place at the beginning of a pandemic to identify people who are at more risk of severe outcomes so that targeted measures can be put in place to help to protect those people. How do these approaches compare? Well, there are benefits and disadvantages of each, but if we look at the target population that is being approached, under the direct strategy there are multiple groups who are at risk of severe disease and they need to be identified, and it's a little bit more complex. With the indirect strategy, we just need to focus on high transmitting groups, and children of school age are recognised to be high transmitters. The location of delivery for direct strategies will be more diffuse. We're waiting for people to attend primary care or calling them into special clinics. For the indirect strategy, schools can be used as the place to immunise large groups of children at one time. Based on this location, delivery is often more timely under the indirect strategy. It can be focused over weeks in schools as opposed to direct strategies which deliver vaccines piecemeal over weeks or months. And the big driver for the UK using this approach is that typically for seasonal influenza, as we said, about 30% of people actually at risk will take up the vaccine, but the indirect strategy has coverage as high as 60% in the UK experience. What happens if we need 2 doses of vaccine? So we said that sometimes only 1 might be needed to achieve high levels of antibody, but in other cases 2 doses are required. This means that we're going to need twice as many vaccines to immunise the same number of people, or else halve the coverage. That means there'd be twice as many clinical school visits, and based on our experience, we know that less people will actually turn up for the second dose, so the coverage of that full vaccination is likely to be less. Full vaccine responses are only achieved after a course of immunisation is finished, and if that requires 2 doses, it's likely there'll be a delay of at least 2 weeks to be directly protected. This means that indirect vaccine protection effects will also potentially be reduced or delayed. We have also observed for some vaccines that the safety profile might differ for the first and second dose, and again this means we have twice as many encounters to record and analyse, and if the safety signal is only present following the second dose, there could be a delay to recognising this. Because of this interaction between direct and indirect effects, it can be tricky to predict what the need for 1 or 2 doses might mean. This figure tries to represent that by showing the results of a simulation in which individuals were immunised in a population at the time of a pandemic event. The far left column looks at the situation where only one dose of vaccine is needed for full protection. The second column looks at the case where 2 doses are needed for full protection, and the number of vaccines is unchanged, meaning that half of the people in the population are fully immunised with 2 doses in this example. On the far right, we take the same number of doses where 2 are required for full protection and administer just 1 to a larger number of people. The height of the bars reflects hospitalisations in the population if the vaccine is given, compared to the case without a vaccine. So the lower the bar, the better the outcome. And here we compare 2 strategies for vaccine: a high-risk strategy, providing direct protection to people we know are at more risk of severe disease, and a primary school strategy, looking at immunising school children as a way of protecting the whole population. We can immediately see that if only 1 dose of vaccine is needed, we have the best outcome for the population program, and the primary school strategy works a little bit better. If we actually have to give 2 doses of vaccine, we still get a pretty good result, but again, the primary school strategy is better. Perhaps a bit surprisingly, if we give only 1 dose of vaccine to more people, our program works a lot less effectively. And here we have the highest incidence of disease, and again some benefit from the primary school strategy. But it can be seen in this particular case that it's going to be more effective to fully immunise the few rather than partially immunise the many. In summary, we know that seasonal flu vaccines are moderately effective at preventing infections. Pandemic vaccines will be modifications of these vaccine types. Early trials will enable prediction of likely protection based on antibody responses and also tell us how many doses of vaccine will be needed. However, close ongoing monitoring in the population will provide better evidence of effectiveness and safety. We've seen that different vaccine strategies could be used to protect the population: direct strategies targeting those at greatest risk, or indirect strategies that aim to reduce infections overall. And we've also shown that what happens when 1 or 2 vaccine doses are needed for full protection. A 2-dose requirement certainly increases complexity of the program, but it may be more effective overall in some cases. And so decision makers will need to decide on whether it is best to provide less protection for the many or more protection for the few. Thank you for your attention. Are there any questions?